This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 999, recorded on April 7th, 2023. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello, Vincent. Yesterday was a remarkable day in Earth history in that the temperature hit 85 degrees in Fort Lee, New Jersey.
Wow.
And that actually should have never happened. Today, it's a little bit cooler. It made sleeping last night very interesting. And the weather outside right now is typical changeover spring weather.
14C.
I don't even know what the temperature is.
So we have 14C.
Oh, good.
And there's a red flag warning.
Probably thunderstorms.
It says...
Not probably thunderstorms? What's a red flag, Alan?
Don't
fly.
Let me introduce Alan so he can tell us. Also joining us from Western Massachusetts, Alan Dove.
Good to be here. Here in Western Mass, it's 50 Fahrenheit, 10C. We also have a red flag warning. That is for dry conditions and high winds, fire weather.
Oh.
Okay.
So it's wind is northwest at 21, gusting 29 miles per hour, and supposed to be gusting into the 30s. And everything's dry enough that apparently it can catch fire pretty easily.
Right. Because at the ocean, a red flag, you can't go in the water.
I think. Yeah, that's like swimming, or you can have a small craft advisory for boats. But
okay. Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 49 Fahrenheit, 9 Celsius. Perfectly sunny, clear sky, and it would be a great night if they had sent me an ISS alert, but I didn't get one for tonight.
Right.
They usually send them, and they're going to be cloudy nights, so I don't bother.
Also joining us from Madison, New Jersey, Brianne Barker.
Hi, great to be here. It is also 14C, 57 Fahrenheit here, and it's getting ready to rain.
Oh. Is that a blue flag, Alan?
that'll put the fire out.
Yeah, exactly.
It will put out the fire. That's right.
It's nice to be in the incubator at any weather. It's always
It's
nice.
always the right temperature in the incubator.
I like it here very much. It's a wonderful environment. And
It is. I agree with you.
Today I just put up a new soft box. I don't know if you noticed, but
You look very soft. Yes.
I look very soft. So I have these round soft boxes that kind of splash on me and on the background. And I noticed when the documentary people were here, they had a rectangular soft box. So I bought it. It was only $30. And it took me an hour to assemble it. It's really hard to assemble, but it's right above and it's shining down. And so there's less splash on the back here. I'll show you how it looks. See there. Background.
How's the heat factor?
And then there's a little bit of a fill light, very, very low otherwise it would be too dark on my face.
So for those of us who don't know, can you explain what a soft box is?
Oh, yeah. So lights these days are LED lights, they're basically circuit boards that glow when you put electricity through them. They don't generate much heat. They're called chip-on board, COB. And they're very intense. And so to make the light soft, you have these big, they look like umbrellas, except they're curved. And then the front has a white piece of diffuser cloth on the front, and you attach that to the light. If I had another camera setup, I could show you.
The larger the light source, the softer it appears to be.
Yeah.
So this is why on a clear sunny day, shadows are sharp, but on an overcast day, shadows are diffuse because the sun is being diffused across a larger area. So a softbox emulates that. It's actually, they've been around since well before LEDs. These were used, similar things were used with halogen bulbs. But the ones with LEDs catch fire a lot less, and they use a lot less electricity, and they don't get as hot.
Yeah, so no more bulbs that get really hot. These are great, yeah.
Okay, I just Googled for the image. So are yours on stands, or they hang from the ceiling?
No, they're on stands. I have a drop ceiling, so there's no way to hang it. I wanted to actually remove the drop ceiling and
put in
put
light
in
bars.
proper rails and stuff, and the landlord freaked out because he
Really?
didn't. Because when you leave, I have to pay to put them back.
What do you mean when I leave? I'm not leaving.
Well, I guess everybody leaves, right? So these are on stands. They're called C stands, and they're very heavy metal. And I have 4 or 5 of them. So I wanted to experiment because the documentary guys got this very cool image. Of course, my background was totally dark. It's like, who was that interview guy who used to sit in front of a black curtain?
Several.
He was good with scientists, right? You know who I'm talking about. Charlie Rose.
Charlie
okay.
Rose.
Right, he'd sit with a black, and so that's what it looks like. Anyway, I think it does look different, and I like to experiment. That's why I like it here. I can experiment and try different arrangements and see what works and so forth.
And LED lights don't give off a lot of heat either, so that's a good factor.
They're very good. You could have a whole room full of them. And now I also wanted to point out that I never know what finger. That is the Luke Jerem glass sculpture that you guys gave me for episode 600. I brought it in this week because I thought it belonged here. And say there it is. It's sitting on a little LED.
Awesome.
It's sitting on a little
Beautiful.
LED that can be different colors. So isn't that cool?
Yeah.
Yes.
Yeah.
Yeah. Very much.
And in packing it back in the original box, which is this incredibly handmade wooden box, I found a note from you guys that I'd never seen before.
Well, I hope it was
Well,
positive.
congratulations on TWiV 600.
600. It's not here. There was a note from you guys that he or his assistant wrote, and then he wrote a little note himself, Luke, saying, "Dear Vincent, congratulations on this amazing science, blah, 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 that you have done all these years." Luke Jerem, he signed it. And
Nice.
the sculpture is called Untitled Future Mutation. And I brought it on the train this week, and it has a big sticker on the front that says Untitled Future Mutation. I said, is anyone going to give me a hard time with this? So,
I'm glad I didn't have a biohazard sign on
Right.
it.
If anybody asks, just say, "It's okay, I'm a virologist."
That's
Oh,
right.
yeah,
that's
that'll
right. Trust
assure
me,
people.
trust
Yeah,
me,
oh,
You'll
no.
get your whole
biologist.
car to yourself. It'll be great. Till the
Exactly.
police arrive.
Have I mentioned the time that I went to a basketball game at Madison Square Garden and they really didn't like my virus keychain?
Really?
Kidding.
Oh.
Yes. Oh no, I have. So
Didn't like your virus.
I
You have the
have-
herpes one?
I have the herpes one.
Oh.
yeah.
Like this, and they
did they
took
think it
my
was?
keys when they were going through things, and they said, "What's this?" They thought it was some sort of weapon, and
It looks a little like an old-fashioned mace or something.
yeah, and I didn't think anything of it, and so I said, "A virus," and that did not make it better.
No.
No,
So
but
what
then
happened?
you clarified and said it's a herpes virus.
Oh, yeah.
That's right.
No, this is not helping, is it?
Yeah, I sort of stood there looking confused at why he did not like my keys, and finally he's like, "All right, fine, whatever, go."
Funny.
Anyway, that's I should do a tour of the video tour of the studio because it's pretty cool, all the stuff we have here. And you'll see,
It's a
Kathy.
lovely
Everybody else has
place.
seen it. Yeah, Dixon and Alan and Brianne have been here. Rich has been here. Angela, of course, has been here.
It's very comfortable.
So it's very comfortable. The furniture's great. And
But I'll be the first one to see the soft boxes.
Yeah.
you'll see the new
Yes.
softbox. Yeah, you'll see the old ones.
Yes.
The old ones are round, and the new one is like a foot wide by, I don't know, 5 feet long. Only because the documentary guys had one. Except theirs was
Light envy.
I took a picture of theirs and I looked it up. It was $3,000.
Oof.
Whoa.
And this thing is well, the light alone was just $250 and the box was $30 because apparently, for film, they want you to be able to customize the color
color.
temperature
Yeah,
precisely.
Of course.
And you have to buy. This weird thing was the actual. So these the light that I have is a round light with a power supply in the back. But the newest thing now is to have a flexible mat with LEDs embedded onto it,
My.
and you could twist that in any way you want, or you can put it in a soft box. And I think that was only $1,200. But the power controller to run it was $2,000.
Jeez.
I mean, why? Why?
Well, because they probably has a whole computer system in it to turn on particular LEDs at particular levels to get exactly the color temperature
Yeah.
you dial in, Which,
Yeah, yeah.
if you're a film crew, you need to match your color temperatures on your lights, and that's very important. But if you have a studio and you're doing, a video for internet release,
Doesn't
you don't
matter.
need quite all that.
Well, there's
No,
something else to consider too, and these devices are very useful in running grow lights for greenhouses
Yeah.
and vertical
Well,
farms.
okay,
I'm
yeah.
sure. Yeah. No, they don't
So-
generate a lot of heat. Anyway, yes, Kathy.
I was just going to say, those people who have been in the habit of fast forwarding because they want to skip the weather are just going crazy
Yeah.
now.
Yes,
I'm
they
sorry.
are. Yes, they are.
Somebody will put
So-
in timestamps about
Yeah,
this, and that you
that-
can skip forward to.
That would be my friend Doug. So,
Yeah.
Doug, enjoy this.
Well, some people like these diversions.
This is a
Yes.
vintage TWiV digression.
This is who
But
we are,
the-
okay?
It's too
The
bad
doc-
it's not stump grinder season yet.
The last comment, this documentary crew, I was amazed at the stuff they brought. Right, they filled the other room with equipment. They had these carts that they rolled in, and everything possible, and all kinds of fancy equipment. The cameras they were using were $200,000 cameras. You have to rent them because you can't buy them. Amazing lenses, a whole bunch of different lights. They would try stuff, it didn't work. They would put it away. It took them 4 hours to set up before I even said a word.
Can you
And
remind us of what the documentary was about?
It's a COVID documentary. Yeah,
I
I'm not
see.
supposed to say too much about it, but
Okay.
I see.
it's being done by a production company in the UK, and it's going to be released in about a year. Netflix, Amazon, Hulu, all the big places.
Oh,
It's going
wow,
to be
that's great. Vincent,
released
that's great.
on, and they had me sit here and be like I was doing TWiV and go through all kinds of announcements and stuff, and they would do the clapper thing, and then
Right.
Oh, yeah.
action, and then the
Do you have
director
makeup?
was standing there, and he goes, "No, Vincent, more enthusiasm, do it again." And he kept doing it over and over again. It's like 20, 30 takes, and then they would
Oh,
put different cameras to do the same thing over. He said he wants me to be the thread throughout it that kind of
oh,
anchors it,
nice.
right? And so he said when we premiere, you could come to the red carpet thingy.
Nice.
So anyway, it's about a year. It was a lot of fun. I learned a
It's
lot.
being
He told me
opened
to
in Wuhan.
he told me to put the desk farther away from the wall to kind of give it some depth. He said, put some equipment on the shelves that's kind of reflective of what you do, besides viruses. A lot of good. And they were actually flattered that I asked them, what do you think I should do with this? Right. So it was fun. Okay. So today we have a lot of stuff, and we're already 20 minutes in. So Kathy, what's up with ASV?
Well, the website is asv.org/ASV2023. We've talked before about ASV Cares grants that members can apply for, and also members can take advantage of the Kitty Corp on-site daycare. So you just go to the site and you'll find the links for that. If possible, sign up by April 21st. We're trying to make sure that both of them can be a go, and we need people to sign up for that to happen. We've talked about other things like satellites and so forth. We can talk about those another time, but I wanted to tell about two additional things. One is that during the dance, we're also going to have playing for those people who don't want to dance. And dancing's going to be great. It's going to be outside. It's in Athens, where Athens is a great music town. So Mark Tompkins, the local host, has been auditioning bands or looking at all their tapes and trying to decide. So it's going to be really good. But there's going to be a virulent documentary. So this is a documentary that was made. It includes Terry Dermody, John Williams, Paul Offit is in it. I haven't watched the whole thing, but they wanted to squeeze it in somewhere. It's 90 minutes long, and we don't even have 5 minutes to shoehorn in anything into ASV. So we said, okay, we'll do it during the dance. And if people don't want to dance, they can go into the
They can go watch a
air-conditioned
movie.
theater. Right. So that's what we have. And then also there's going to be AthFest going on. And AthFest on Friday, Saturday, and Sunday is sort of a local music event in the downtown area. A couple of the streets will be blocked off, and several music stages and so forth. Of course, you should be at the ASV meeting, but if you get there Friday, you can certainly go to AthFest and check out some of the music.
Get your
that
ass
night.
down there.
Yeah.
Cool.
Right.
I'm all booked.
Good.
I better do it. I haven't even registered yet. I better get going, get
Yes.
the early bird.
Yes.
Okay. Before we do some papers, I want to point out an article that Daniel actually talked about briefly on his clinical update this week. This is a stat article entitled, "If We Don't, Others Will, White House COVID Advisor Calls on Doctors to Combat a Vacuum of Medical Information" by Elizabeth Cooney. Ashish Jha, coordinator of the White House COVID response team, who has, I think, said pretty good things overall, right, about the pandemic. But, asking doctors to comment about virology is a little suspect. But the problem here is that, what he says is correct. He says what we have seen is the widespread propagation of misinformation and disinformation. And the reason it's taken root is because there's an information vacuum. And I just got burned up over that statement.
Yes, you should.
Because we're working hard as hell. We've been doing this for 3 years. We have over 1,000 hours of COVID programming. And this guy says there's an information vacuum. I agree we need more people filling in a vacuum, but there's not zero, Jha. Come on. So I put a comment at the bottom of the article. I wrote, somehow Dr. Jha has overlooked the 1,000+ hours of COVID programming on This Week in Virology, a podcast with virologist and an infectious disease physician. While there is no doubt the misinformation misinformation is rampant, some of us are trying to counter it, and Dr. Jha should know about our efforts. So I think a guy of that prominence should know this, that we're working hard.
The problem nowadays is never an information vacuum. We are flooded with information. The problem is what information gets brought to the top of the majority of people's feeds, for want of a better term. Whatever it is that they're staring at in these glowing rectangles that dominate our days. And they're not staring at TWiV. I mean, the 50,000 or so people who subscribe to us are, but that's a drop in the bucket. And the problem is that what comes to the top of Facebook and Twitter and Instagram and TikTok and all that is more likely to be the misinformation and disinformation, because people will be more likely to keep scrolling that. And that is the sole measure of profitability for the social media platforms, unfortunately.
Sure. Yeah.
How do we get Daniel Griffin's visibility to this person?
I think that we should. I mean, he's talking to a group of physicians, right? That's
but
what
there
this
is.
is directed at.
He does make a very good point, which is physicians hold a unique spot of credibility, and they are. Everybody has or should have or knows they should have a physician in their lives, and they will see that person occasionally. And there's ample evidence from decades of research that that physicians, specifically, MDs, not the first nurse you see when you walk into the hospital, but the doctor, is the most credible source of medical information for most people. And a doctor saying you should get vaccinated carries an enormous amount of weight.
Right. The problem is many doctors are making mistakes. They're giving antibiotics. They're saying there's Paxlovid rebound, which he points out there's no Paxlovid rebound.
There are a lot of pressures on doctors to do a lot of things, and there's a lot of time pressure. They can't necessarily communicate about virology with somebody who came in for their heart checkup. But yes, this is the front line.
Look, we get doctors emailing Daniel every day. What do I do with this? What do I do with that? Okay.
Yeah.
Because they don't know, and Daniel can help them.
Yeah.
And so, I don't disagree that there's a vacuum and that doctors have to fill their part, but there's also what we and others have been doing. And I think many of the doctors have caused problems, like cardiologists and others who don't know about viruses. So I have a really mixed feeling about this. That's why I brought it up. And so put the link in. You can take a look at it. I called this rant. The subheading was rant.
Right. We have a rant, a snippet, and a paper in this episode. That was the rant.
Okay, the paper is interesting. Well, this is a preprint, although Kathy points out it's now in Nature. What a quick review.
Mm-hmm.
I wonder if they read it.
Well, it's a preview. And it could be that they just put up the preprint when they knew it was going to be, when it was accepted.
Yeah.
But,
So this is a China XIV. There's no R in there.
China
bioarchive,
right.
medarchive, they all have R's in there. But I guess this is just China XIV. So they're trying China Chive,
yes.
maybe. Yeah, I guess they did it on purpose, right? But anyway, it's their preprint server, and this is surveillance of SARS-CoV-2 at the Huanan Seafood Market. This is, I guess, the data that they partially put out a year ago, and then a couple of weeks ago they pulled the sequence off of GIS8 as we documented 2 weeks ago. A number of individuals then published a summary, including Florence Desbarres, who first found animal sequences in SARS-CoV-2 positive specimens. And so now the China CDC and others have published their version of what they think is going on. And so this includes George Gao, who was the former head of virology at China CDC. Virology or the whole thing? I don't remember. Does anyone remember?
I think he was the head of the China CDC, wasn't he?
The whole thing?
That was what I thought too.
Somebody Googling?
I'm looking in the paper for the, well, that wouldn't necessarily
his current affiliation is not necessarily.
Right.
He's at the Chinese Academy of Sciences now. But this is China CDC.
Right, so this is a paper from William J. Liu is the first author. Guizhen Wu is the last author. There are 1, 2, 3, 4, 5, 6, like
7 co-first authors. Co-first authors.
Co-first authors.
So just to be clear, the Huanan Seafood Market is in Hunan.
Right.
Huanan. This is Huanan Seafood Market
Wuhan.
in Wuhan. Yes,
Wuhan.
it is not
I meant to say
Hunan.
Wuhan.
Yeah.
Yeah. And it's China CDC, University of Shandong, Shanghai Jiaotong University, Institut Pasteur of Shanghai, Institute of Chinese Academy of Sciences. So we have addressed this situation the last couple of weeks. So last week we talked about China re-releasing the sequence data from the market, and then the week before, I think we talked about the document on virological.org, which talked about finding various animals, non-human animal sequences in SARS-CoV-2 positive samples from the market. And now, so this is what China has to say about it. And their conclusion is, this study provides convincing evidence of the prevalence of SARS-CoV-2 in the Huanan Seafood Market during the early stage of the COVID-19 outbreak. That's a very muted conclusion compared to...
Yes.
Yeah.
Because officially, the Chinese government is still trying to push the story that the virus originated, spilled into humans outside of China, or at least originated outside of China.
Well, there was an
So this is
remark where they said that actually.
I kind of see this as that this paper was pried out of them because the analysis had already been done and published by another group, and no sense closing this barn door. So here are the data, and they're still trying to advance this idea that frozen foods, cold chain products as they call them, were contaminated with SARS-CoV-2, and that was somehow the source that brought it into Wuhan, which is about, I think that's maybe about the same level of probability as the lab leak nonsense.
Well, there's no evidence for it either,
There's
right?
no evidence whatsoever to support that, and then a lot of reasons to believe that that's a very poor way to transport this virus, and yeah.
We publish sequences the same as their original withdrawn sequence?
I think they are. Go ahead, Kathy.
Well, actually, I don't. They don't. Dixon, they don't publish the sequences in the paper, and whether what they have uploaded is the same as now as.
Didn't anybody grab it in the very early time?
They
and then
did,
keep
and
it?
so I would assume someone has actually looked at that. I just don't know the answer.
Right.
Yeah. So
I would like to
since
know
they
that.
re-uploaded the sequences, people have been looking at it, but haven't heard anything.
I want to make a point now that the preprint that some of us are working from is different from the Nature accelerated article preview, which For instance, it starts off, you'll be happy about this, Vincent, because the paper abstract starts off, emerging in December 2019, coronavirus disease 2019, COVID-19, eventually became a pandemic. Whereas in Nature, it starts off SARS-CoV-2, the causative agent of COVID-19.
An editor was involved in this.
Exactly.
Good. Very good. Glad to hear that. Yeah, I just saw that Nature link recently.
Yeah.
So I'm working out of the preprint.
Yeah.
Okay. Anyway, so what do we have here? They have a little description of the market, the Huanan Seafood Market. I should still remember in our first episode in January 2020, where is this market? We're trying to Google it while we're talking about it. Anyway, it is in Wuhan. It is a 50,000 square meter market with 678 stalls and located near each other in an extremely crowded condition. They write that. There are east and west zones, and seafood and animals mainly in the west zone, livestock, meat in the east zone.
And this is 800 meters from a major train station, so it's a busy place.
Yeah, and so among these stalls, 10 domesticated wildlife animal selling stalls were identified. These are in the southwestern corner of the market, which we've talked about before. That's where the animals that are susceptible to SARS-CoV-2 were raccoon dogs and civet cats and others. That's where most of the SARS-CoV-2 positive environmental specimens came from. There's a nice heat map in the virological.org paper picture of that, and that's on the thumbnail for that episode of TWiV as well. All right, so according to records during late December 2019, animals and animal products were sold in these stalls, and Eddie Holmes went there a few years before and took pictures of them. And what kind of animals? All kinds, including animals that are not important, like snakes and birds of various sorts, deer, badgers, rabbits, bamboo rats, porcupines, salamanders, crocodiles. Oh my gosh, all these animals that people want to eat.
Bats.
They don't actually mention
Right, they mention in the
bats.
paper, so in addition to the animals being sold, there were stray animals. And so that's sale records of late December 2019. They shut down the market January 1st, 2020.
Right.
And then the China CDC sent an epidemiological team there the morning of January 1st, and that's where these samples start.
Yeah. Now, they say we sampled until March, but this thing was sanitized very early on in January.
Yes.
So, they killed all the animals, burned them, they washed the walls and everything down. So I'm not sure how much was left by the time they got there, right?
Yeah, something made me think that they did have some stray cats wandering in and snakes evidently too, but
snakes.
Yeah.
They have a peridomestic
ecosystem. They swabbed the 10 cats and a stray dog that wandered around. And they swabbed all the surfaces.
Have any of you traveled throughout the Southeast Asia or Asia areas and seen these live markets? I have.
Okay.
I went
There
to.
is a peridomestic ecosystem.
Yeah, I went through a live market in Guangzhou.
Yeah, well, people take them for granted, and they probably even end up harvesting a lot of that meat when nobody's looking.
All right, so they have 923 environmental samples and 457 animal samples. They swab doors and corridors. The outside surface of the rolling shutter doors of the stalls and the corridors were disinfected with 1% bleach throughout January and February. Goods were cleared and disinfected until March. So of these 923 environmental samples, 73 were positive by nucleic acid amplification. And the cycle thresholds, which means, how many cycles you need to get a signal of background, range from 23 to 41. So the higher the number, the less nucleic acid there is. So that's not a lot, but these are environmental samples, right? Of the 828 samples inside the market, 64 were positive. And, these are sewerage wells and floor swabs,
surfaces,
and so
floors,
yeah, surfaces,
and a couple of pieces of machinery.
And of the 64 SARS-CoV-2 positive samples inside the market, 87%, that's 56 out of 64, were collected in the West Zone. So the West Zone had the preponderance of positives. You know, I mean, if someone, I guess someone, because they're arguing someone came in and brought the virus there, and then it amplified, and it seems to me it would amplify all over the market if that were the case, but who knows?
Does anybody have any data on the daily use of this market?
I'm sure that it exists,
population-wise.
I'm sure it exists.
It probably was in one of those early papers. There was that paper that talked about the animals in the market for sale in end of 2019. That was where they had studied that for some other purpose.
Right, the wildlife paper.
Yeah, the wildlife paper. So I wouldn't be surprised if, for example, those data are out there. I've also been looking online to see if there is any discussion of whether or not the data changed when it got reposted.
Yeah.
And
yeah.
I do not see any discussion of that. There's other discussion, but not discussion of that.
I'm pretty sure there'd be discussion of that if somebody had found
exactly.
it. Yeah,
there is additional analysis of the data, so it looks like people are looking at it and finding it to be the same.
Okay, let's see, 40 positive samples, 44 were among 21 vendors, 19 of whom were in the West Zone. So most of the positive samples in the West Zone, and that's where the live animals were. So that makes sense. And they say the positive, they say no differences were observed between different vendors, including poultry, cold chain products, aquatic products, livestock, seafood products, wildlife, and vegetables, which is very strange, right?
I don't know how big the statistical base was to make that argument, because the spreads on those data are huge.
Yeah, they are.
So I think we're dealing with small numbers. In fact, if you look at the numbers, it's like poultry 8 out of 37. Cold chain, 16 out of 87. And they say, I think elsewhere in the paper they mentioned there's overlap here. So somebody might be selling poultry products and frozen foods and, some other things, which makes sense. If you've been through any kind of a market, you see multiple things being sold in a place. So I don't think we can read anything from this.
But they want to make this. Their agenda is that it was everywhere. And
the goal of the paper is to try to say it was everywhere, when in fact, I mean, the data pretty clearly show it was in the area where the wild animals are. And
so they conclude.
A lot of reasons we're not listing all the animals is that many of them are on the endangered animal list, and they're illegal. And they don't want them to know that people are out there collecting the natural wildlife and bringing it to a market for sale almost on a daily basis. I didn't see the word pangolin. I didn't see soft-shell turtle. You don't see any of the animals that the World Wildlife Association has put out an all-points bulletin on. If you find somebody capturing this animal, report it to us and we'll take action.
You may remember we did a paper last year of a guy who'd done a survey a couple of years earlier of this market for other reasons, and he cataloged all the animals that were there. There were no bats and no pangolins, right?
Interesting.
But there were plenty of other
species
things that ought not be
like
there.
Sumatran tiger.
Oh, yeah.
And
plenty of other things that were officially not there were in fact there.
Yeah.
That's the point.
And like raccoon dogs and civet cats, which we know are susceptible to infection. So their conclusion is the virus may have been circulating in the market, especially the West Zone, for a while in December 2019. Really might have been. I mean, leading to an extensive distribution which might have been facilitated by the crowded buyers and the contaminated environment. Well, what about it came from an animal and got amplified? What about that? Oh my gosh. Anyway, so this is the part that I had a lot of trouble with. They have 457 animal samples from 188 individuals belonging to 18 species. And the sources are unsold goods in refrigerated and freezers and goods kept in warehouses elsewhere. 3 Chinese salamanders, which are found in a fish tank, were swabbed. As Alan said, 10 stray cats, 27 cat feces, 1 dog, 1 weasel, and 10 rats. These are stray animals who now know they shouldn't have gone there.
And a partridge in a pear tree.
All these animals were negative for SARS-CoV-2. And I would say, yeah, they're not the right animals.
Yeah.
That's right. That's right.
So the right animals were all burned, right?
Indeed.
So that's the problem. We don't have any of the susceptible animals. They were gone. But we have all this environmental stuff that we know is a mixture of human and non-human animals.
And they go through, I mean, they go through the various genera that were present in various places. And it's stuff you'd expect. And also, Nycterudes, which is the raccoon dog, Atelorix and Erinaceus are hedgehogs. And those were concentrated in the area where the positive samples were. There was one thing that leapt out at me, though, that is completely... They just tossed this out there. Top abundant mammal genera of the defeathering machine sample was Canis.
Yeah, what's a dog?
Okay,
Maybe...
so why is there dog DNA on the defeathering machine? Can someone explain?
Well, so I put in the Ars Technica article, which you might not have had a chance to read yet. It's not very long, but they talk about the fact that when the group that was not from China evaluated the data and they first turned up things being in Canis, they also thought it was just ordinary dogs until they realized they weren't using the right database to search against. And when they used the right database, then it came out raccoon dogs.
Okay. That still doesn't explain why they're on the defeathering machine. I'm assuming that's cross-contamination from an adjacent machine or some other operation that was near the defeathering machine. I don't think they were actually defeathering.
I can imagine that nothing goes to waste in the Wuhan wildlife slaughtering market. And whatever you don't buy is already eaten by some other group.
Yes, and as Kathy pointed out, during the brief period I was just trying to do that quick search, the number of things I did find about potential misidentification that tie in exactly with that article that Kathy posted was not 0.
Yeah.
Right. So this could do more damage than damage control for those in charge of the epidemiology and public health aspects of this infection in China. This is not a very favorable article in my view.
They did isolate infectious virus from 3 specimens.
That's nice.
They put them on cells. They got CPE. They took pictures in an electron microscope. You could see coronaviruses. And these were samples with low CT values, right? More nucleic acid. 2 of them were samples of the ground, and 1 was a sample of the wall. Okay. So let's see. What else is interesting? They got 7 complete or near complete genomes. And the ones where they got virus were identical to the reference strain, reference Wuhan HU1. They also see both lineages, right? There are 2 lineages of SARS-CoV-2 circulating early, and they find them both here in the market. And this was actually in the preprint a year ago, and Eddie Holmes thought that was a big deal.
Yeah.
That they quietly published this, showing that both lineages were here, which is consistent with multiple spillovers, right?
Right, but they somehow kind of discount it.
I know.
Yes.
Yeah,
they talk around it in this paper, but it's virologically, it's really hard to explain 2 different lineages happening to be on, I don't know what they're saying, people coming into this market at the same, no.
Yeah.
I think this is where it happened.
Almost none of the data actually contradict sort of some of the things that we've seen before from say the Warobi paper or things like that.
I mean, we know this happened once before in China in a market, right?
Yes.
So there's an obvious thing here, and they're trying to fight it. And
well, that's the problem. The last time it happened for SARS-1, they lost over $60 billion worth of tourists.
Mm-hmm.
And so they're not going to do that again. I was involved in that peripherally. Our School of Public Health was invited over to China to help. the Chinese Department of Public Health reorganize. And we were there for 3 days. We went home. And the end result was that they fired virtually everybody that was on the Chinese Department of Public Health and hired all new people.
Hmm.
There wasn't one person that was the institutional memory to carry over to the next group to advise them as to what they did. So this is, obviously they don't want that to happen again. But it did happen again. Even if they didn't want it to happen again, it happened.
So
they shut down the whole country.
The next part, they look at mammalian DNA in these specimens. They said you could find mammal species in most samples. 70 different genera were all identified in all these environmental. 70 different mammalian genera.
That's incredible.
And
are there 70 genera?
Apparently so.
Oh yeah.
No, I was just joking.
So I'm not sure when to jump in with this, but
anytime.
I really hope we can post this Ars Technica article also. It's written by Beth Mole, who has a PhD in microbiology. Anyway, the genetic flaws don't appear to end with the misidentification of raccoon dog genetic material. According to the Nature study, the China CDC scientists also found traces of things like the highly protected, generally large giant panda, bears, chimpanzees, gray seals found in the North Atlantic, and rodents from the Chilean Andes. It's unclear why the Chinese scientists did not correct their analysis, but now that the metagenomic data is public available, outside researchers are keen to dig in.
So is
it
reasonable
there wasn't
to
actually
speculate?
panda at the market. Pretty sure of that.
Oh,
That is a misidentification.
Someone had a pet
of the
panda.
DNA
They brought him to
sequence.
work every day, and that's what they're.
Or North Atlantic
That's
gray
right.
seals.
Well, they were, I don't know. Is it within the realm of speculation reasonable to assume that this may indeed have come from someone who actually works in the Wuhan market?
What do you mean comes from someone?
Animals come in, there's spillover, it infects one person, and that person becomes the node
that's
for
what we're
infection
thinking,
for
right?
everybody else.
Well, right.
That's
so
what I'm thinking. That's what I'm thinking.
I'm thinking that the animals brought to the market were carrying probably through one supply chain of, say, raccoon dogs, were carrying the two lineages of SARS-CoV-2. It spilled over into humans at or adjacent to, like while they were trucking them to the market or at the market. But I don't think... I mean, I guess it's possible that the person who's driving the stuff to market could have gotten infected with one lineage, and then somebody who's driving a similar load could have gotten infected with the other lineage. The two lineages really complicate putting it any place besides the market.
Right. And how about the guy who's in charge of killing the raccoon dogs?
Yeah. I mean, that would be a key candidate to...
I wonder what happened to him or her.
Well,
that would be an interesting epidemiology.
So remember, none of these animal specimens have SARS-CoV-2. All the SARS-CoV-2 is from environmental specimens,
Yes,
right?
And the analysis by Debarret and others suggests that many of those had raccoon dog and civet DNA, and some have no human DNA at all. One of them from the west part of the market had SARS-CoV-2 and either raccoon or civet, I don't remember which, and no human DNA. So that came from an animal. And of course, China would argue, a human infected that animal. Which is not what most people would argue, right?
No.
But trying to fit it to their agenda.
Forgot about Occam's razor there.
So, there's no mention here of that specimen, right? That one specimen, which is mentioned in the Ars Technica article, I believe. Let's find that. It's very good. It's very well done.
The 0576, cart Q61. Yes.
And Warraby mentions that, right? I think.
That's the one that they focused on, right? Yeah.
Surrounded by a high density of CoV-2 positive samples, and one was itself teeming with raccoon dog material. They found a sample contained 1,252 genetic fragments with 100% identity to raccoon dog, no perfect matches to the human genome, and the finding hints at the possibility that the virus was from the raccoon dog, not humans.
Right.
Yep.
And the reason we don't have samples directly from the animals is that the animals had all been destroyed or removed.
That's right.
by the time they were doing it, because they shut down the market, which was the correct thing to do, just so we're clear on that. But they didn't get samples from any of the animals that were there.
They should have sampled before they shut down the market.
They should have sampled. They should have gone in. Well, get the people out, lock the place. I guess force the merchants to leave their merchandise in there, and tell them you're going to sample their merchandise and then destroy it.
What they could do is go back into the source of these animals outside the city and sample those.
Although I suspect that source is no longer in business.
Yes.
And is probably not an easy person to find if they follow the news at all.
Yeah.
And they also might be sort of an intermediate between the earlier source of those animals.
Yeah.
If you could have gone at the time, you might have been able to get to that source.
Yes.
And they may be back in business now, but
under a different business name and maybe a different market and
new set of animals.
If that were me, I would not be forthcoming. I'm sorry.
Right. If you put a pin on the market and then kept track of all the human cases after that, how soon after they realized that there was something horrible going on did they realize that it might have been coming from the Wuhan market? I mean, can you go back in time?
Yes. Well, here's a story. The original 40 cases were published in the New England Journal, right? And the first few were clearly market associated. And then a lot of them were not. And so people say, it wasn't the market. But then when Warraby did the analysis more extensively, it became very clear that that was the epicenter very early on.
It looks like a plaque. I mean, Warraby's maps are just absolutely damning that it radiates out from the market.
Well, and Alan, you're implying, I think, or I'm inferring from what you're saying, that if you tried to go back to the source, they know that they were the source.
Well, I
suspect that's the case, that they don't know that they were the source.
They don't know
until
that they
maybe now when they read this and they find that they sold stuff to cart Q61. Then maybe they have some inkling, but
they read this.
I don't think. I don't think that the business or person who sold the raccoon dogs to the market necessarily knows that they are the source, but they certainly know that they're in the raccoon dog business and that they're in the wild animal trade. And as the authorities start closing in on that, I think, that would be the point when most people would find another thing to do and maybe not hang on to the paperwork from the previous thing. Just like everybody in that business would probably want to have a low profile, is my guess.
Yeah.
You bet. Or, even worse.
Or they're just not around anymore.
Exactly.
It's for a variety of reasons. I mean, they could have been carrying an early spillover of this virus, and they may no longer be with us.
Correct. Yeah. I mean, I keep hearing the word, the barn door is open and the horses have gone. That's not what happened. The ark fell upon bad times and the door opened and all the animals spilled out and they all ended up in Wuhan. I mean, what didn't they keep there? Did they slaughter horses there? Cattle? Of various kinds. There must have been.
I don't think those were slaughtered at the market.
Right. Yeah. Right. There's meat there.
Yeah, they had meat over on the east side of the market, which was slaughtered elsewhere. I mean, just like my local butcher shop.
Right.
Which, by the way, sells more than just a couple of species. I can buy meat of quite a few things.
I am certain. It's a zoo. It's an absolute zoo.
What can you get? You could probably get deer and boar and...
Yeah, get deer, boar, bison, alligator. I just cooked a rabbit the other night, and I did not shoot it myself. I bought it from butcher shop.
Close to Easter.
You should try jackalope.
Jackalope. Oh yeah, that's good.
I mean, there's probably at least a little bit of pork there too.
There's quite a lot of pork there. I am a regular patron of their pork section.
But maybe not 70 mammal genera?
No.
Maybe not 70. Okay,
this is an exotic marker. So their conclusion is we need to do more surveillance of wild animals to explore the potential natural and intermediate hosts for SARS-CoV-2. That's interesting. So it didn't come from frozen food, you're thinking, right?
Not Mrs. Paul's fish sticks.
Did that get into the Nature paper, Kathy? It probably did.
So is that the very last?
It's the last
paragraph?
sentence of the last paragraph.
Let's see. That's in fact what I finished reading. See, more work is needed to investigate potential origins,
surveillance of wild animals using a viromic approach.
Yeah.
Interesting. All right. By the way, I have to tell you guys this story because you will appreciate it. All right. I said jackalope because a jackalope is a mythical animal which looks like a combination of jackrabbit and antelope. And it's actually a rabbit with papillomas growing out of its head and face, right? They're really long and they look like horns, antlers from a distance. But they're papillomas caused by a rabbit papilloma, which was the first discovered by Robert Shope, I think, the first DNA virus to cause tumors like that. Anyway, so I did the transformation lecture this week where I talk about that. And at the same time, at the end, I talk about how Henrietta Lacks' tumor was caused by an HPV because when we did the genome sequence, we saw the E6 and E7 oncoprotein genes inserted into her DNA, and that makes the cell divide and divide. And of course, HeLa cells are based on the first two letters of her first and last name. So what am I talking about? Many years ago, I went to my son's high school and talked about human papillomaviruses in the Henrietta Lacks books and HeLa, and I told my son, I said, it's HeLa. So he started calling himself AIRA, A-I-R-A, Aiden Racaniello, right? So this Saturday, he's an amateur DJ, and he's doing a gig in New York City with a bunch of other DJs. And the gig is called the Feast of the Jackalope.
Okay.
And, they have this picture of a jackalope sitting at what looks like the Last Supper table with all these other beasts sitting around it. But his DJ name is AIRA. So I told my class this because there's two connections to my transformation in oncogenesis. Like there's the jackalope and the papillomavirus, and then there's HeLa cells and AI, right?
Isn't
that?
Yeah.
That's excellent.
Good.
But I said, I'm not going because it's 12 midnight to 5 a.m. What kind of a concert is that?
That's
For young
the kind
people,
you would
that's what
have
kind of a
in
concert
Athens,
is.
Georgia. Yeah.
Why does it have to be till 5 a.m.? Don't people have to sleep? No, huh?
No, it's a cheap studio time.
Oh, anyway. So our paper today is also one that Daniel brought up. He just touched on briefly. So I thought we would do a deep dive, Nature Microbiology, live attenuated vaccine SCPD9 elicits superior mucosal and systemic immunity to SARS-CoV-2 variants in hamsters. And, Daniel said, okay, let's save all the hamsters.
Save those hamsters, yes.
Okay, where is this from? Wow, lots of authors. Oh, it's at the end of the paper. Well, the first author is Geraldine Nguyen, and the last author is Jacob Trimpert. I'll go to the end and I'll figure out who's from.
I'm sure we have
The first two are co-first, and the last three are jointly. The paper was jointly supervised by them.
Okay, so Julia Adler is the other first author, and Dusan Kunek is the other senior author.
Okay, wow.
And now Vincent with the affiliations.
My God, there's a lot of figures. Here we go. So it looks like it's from the University of Charité University Medicine Berlin, right? Humboldt University, Freie University. Oh, lots of universities and institutes in Germany, and a little bit of Hong Kong at the end. So mostly Germany. And I thought it would be interesting because, we don't have any infectious attenuated SARS-CoV-2 vaccines that have been licensed. And this is kind of
Not
interesting.
for lack of trying. There were a bunch early on that I guess
Tried.
either didn't pan out or didn't get pursued.
So as you know, an infectious attenuated vaccine is cool because it infects you. It mimics natural infection without making you sick. At least that's the idea for polio vaccine. If you're 1 in 1.4 million, you might get paralyzed, but that's unfortunate. And of course, influenza, flu mist is given intranasally, but that's not a poster child for a nasal spray being a great vaccine because it's not any better than the other influenza vaccines at preventing severe disease.
On the other hand, It's a lot easier to deliver.
It's very easy to deliver. Yeah, you don't need a needle for sure, and that's a big deal.
That is a huge
Yeah.
deal. A lot of vaccine hesitancy,
Yeah.
etc.,
Yep.
in fact, stems from people being afraid of needles.
What was that site, Alan, that had nice pictures of people getting shots? What was the name of that?
I think it was Self Magazine.
I think
a few years ago did a. They said, you know, stock photos about vaccines are horrible, so let's fix it.
Yes, I was looking for that.
They had a bunch
this
of
week.
stock photos of a bunch of different people getting vaccinated, and it's not a huge needle and a screaming child. You know, it's
Okay.
much better pictures.
We should make sure to point out that there are kind of two details that are key about this vaccine. One of them is that it is a live attenuated vaccine, so it is a live vaccine that replicates, just as Vincent said. And separately, this is also a live attenuated vaccine that is delivered mucosally.
Yes.
So there are some other live attenuated vaccines that are injected with a needle, kind of the way you traditionally think of a vaccine. But the key here is both that it's a live attenuated and that it's being delivered to the site where infection might normally happen to try to induce immunity there.
Right.
Brianne,
So that
can
matters
I-
for more than just marketing. It's not just an easier route. It's also the route the natural virus takes, and it gives you.
Yeah, but
exposure.
I'm sitting here thinking what Jens will now write as a result of just hearing Brianne say live.
Yeah, I was going to say, could you say
You
infectious?
should say infectious.
But
Just infectious.
so Brian, the paper we did on immune, they say, you know, when you get an infection, you get that mucosal immunity, which you
Exactly.
don't get from intramuscular vaccines, and so this would provide that theoretically.
Right, exactly.
I mean,
so,
that's the polio story, right?
So one of the two polio
And I
vaccines.
will point out
yeah.
that throughout this Nature microbiology paper, they use LAV, live attenuated vaccine,
Yeah.
as the term. It's the term of art.
It's what it is. We're stuck with it, but Alan,
Sorry,
it doesn't
yeah.
You're not
mean
stuck with it. You
doesn't
can
mean I
change it.
can't complain. Got
Or
a
you can change it,
platform,
just
got to use it. Okay,
I like replicating attenuated
replicating
vaccines.
is good,
Yeah.
beautifully done.
but
For 30
where did it?
points.
Who said this the other day? Oh, I'm not going to be able to remember it, but okay, I'm not going to remember it. Anyway, this is an interesting infectious attenuated vaccine because it's made by codon pair deoptimization. And oh boy, that's not easy to parse. You cannot figure out what that is. So remember, the genetic code, the mRNA specifies amino acids in triplets of bases, right? So that's a codon, 3 bases. And codon pair, it turns out that certain codons are usually next to each other. Many codons can code for the same amino acid, sometimes up to 6, right? And so not all 6 are next to each other. So if you have 2 amino acids, it's not just random what would be next to each other. There's certain codon pairs that are that predominate. So what they do in this codon pair deoptimization, they maintain the amino acid sequence. So alanine, tyrosine, but usually there's certain codons that are next to each other that would code for those amino acids, and they mix it up. So they break the rule, whatever the rule is. So they're underrepresented codon pairs next to each other, synonymous, which means they code for the same amino acid.
So there are
Mixing them up.
multiple different codons that
Right.
can code for the same amino acid. So you can get alanine with a couple of different
Right.
triplets.
But
And different species of animals
And
use
different
Yeah, they're
species
different too.
tend to use certain codons more frequently.
It's
And
right.
so basically what they've done is they've taken the things that are the codons that are usually used quite frequently and switched them to use the unfrequent ones.
but next to each other.
Yes.
Right. It's not just, alanine is coded by this, mostly by this triplet. It's 2 of them next to each other. Alanine, tyrosine would usually be coded for by 2 particular of the triplets, and they mix it all up. And that turns out to attenuate viruses. It reduces their virulence. The mechanism isn't really
It makes them
understood.
less alive.
It makes it
Probably something like the cell has a harder time translating it.
Maybe.
Because it's
Maybe.
a little like, oh, that's a weird way to spell that.
yeah, especially because I think it's related to the host cells codon use.
Yeah.
That's right.
That's right.
It's not just the virus's codon use; it's actually the host cell, because the host cell is the one with the ribosome.
I always looked at this as a redundancy feature of life in general, as a backup in case somebody makes a mistake at some level. There's another thing to come in and take the place of it. Is that a good way to think about that?
You mean having so many codons per amino acid?
Exactly right. Exactly right.
Could be. Yeah. Maybe there are multiple reasons, right?
I mean, Alan, you fly. How many redundant systems are there in a typical...
Oh, so many. Yeah.
Right.
Right. And having multiple codons encode the same thing does mean that if you have a bitwise change in the DNA and in the RNA, that is less likely to cause an amino acid change than it would be if you only had one codon that could do that.
Exactly.
So you have added some robustness to the system.
There you go.
But Alan, you only have one engine on your plane.
The planes I fly only have one engine. Yes, it's twice as expensive to fly with two, and so yeah.
But you can glide with one engine, right?
I can. The advantage to the planes I fly is that they're slow and they have low stall speeds.
Right.
So I can, in principle, land in a lot of little places that would not be. Couldn't put an airliner there.
Like a highway.
Preferably, preferably a nice open farm field. That
that would be nice.
Just
unplowed.
Could you land in the Hudson River, Alan?
Ooh.
I'd rather not, but the ditching statistics for small, slow planes like I fly are a lot more encouraging than they are for airliners.
Oh, that's a fact.
That's why what Sully did was so amazing. But there are many other cases of people flying planes like I fly where they ditch and they're fine because they're hitting the water at 40 miles an hour, whereas an airliner, you can't get down to 40 miles an hour to hit the water.
What was he traveling to?
Oh, gosh, what's the stall speed on it? What were they in, an Airbus?
150?
Yeah, it's faster. And it's also,
It's very
it's
fast,
it's a bigger plane,
especially
with,
if you're in it.
the big engine scoops under the wings that are going to dig in, and
No, he did a great job.
it was amazing.
So
Hello, listeners who really enjoy our banter that's off topic.
Yes.
Today is a banner
We're on
episode for
another
you.
digression.
The thing is, things
Right.
come up, and I'm curious, and I just want to find out, right? What's wrong with it?
Absolutely.
But
Nothing.
this codon deoptimization has been done before for many viruses, and we've talked about it on TWiV many times. There is a great review article by John Coffin. It's called "Attenuation by 1,000 Cuts." I really like that because, in fact, you make thousands of base changes to do this. And so one of the ideas is that these vaccines will not revert. They will not genetically revert because you have so many changes that it's not possible or not likely. Although we did a paper on TWiV showing you can get massive recombination and restore function. Anyway, so polio, it's been done, respiratory syncytial virus with dengue, and now SARS-CoV-2, there are codon deoptimized candidates. And that's what they use in this paper to immunize hamsters. And they're using Delta as the antigen in the challenge virus. And they're comparing this codon deoptimized virus, SCPD9, let's call it 9. They're comparing it to the Pfizer mRNA vaccine and then add 2 spike. So it's an ad vector carrying the spike gene. These hamsters get a single dose or prime boost, and then they're challenged 21 days post-vaccination.
And they did the prime boost in different combinations, so they could get either 2 doses of the same vaccine or 1 dose of one vaccine and 1 dose of another vaccine.
So let me just say something up front, okay? 21 days, you're going to get great impairment of infection because the antibody levels are high, T cell levels are high, okay? And you can make a paper that says, oh, this attenuated vaccine is fabulous. But why don't you do the experiment 3 months later and tell me how fabulous it is? Because I'm guaranteeing you that the mucosal antibodies are going to go down quickly, right, Brianne?
That is the idea.
Yeah. So, to say that, I mean, the press picks this up as, oh, nasal vaccines are going to block infection, they're going to block transmission. Yeah, if you do it 21 days after vaccination, but if you do it 3 months or 4 months or 5, no, it's not. It's not. So in this paper, they don't even mention that the durability, right? And they just say it's great and it's better than mRNA, which I'm not surprised, because when you put the vaccine in your shoulders, not a lot ends up in the nasal cavity. But so that's a limitation. I think we're going to have to, if this makes it to people, and it may, they're going to have to see, in 3 to 6 months what goes on, and I'll bet they're going to be infected. It's probably not going to blunt transmission very much. Anyway, I wanted to say that in case I forgot it later.
Yeah, there's another limitation I just want to put right up at the top, which is throughout this, I kept thinking, well, it's hard to find any humans nowadays who don't have multiple vaccine doses already on board and or multiple infections with this virus. And one of the things that we're going to talk about extensively is the mucosal immunity that this induces, which you also get from infection. So if you got vaccinated and then infected, which is the right order in which to do it, by the way, then the infection is going to finish giving you the mucosal immunity. So then what are we accomplishing by giving this attenuated vaccine?
It's hard to do it right now. You're absolutely right. And I often say that, and people say, you're being negative, saying we can't test another vaccine. But the reality is, at least in many countries, a lot of people have been immunized and infected. Not everywhere, though. Africa still has very low coverage of vaccines. So
and certainly a nasal vaccine is going to be easier to deliver.
True.
How long does the
Kathy?
I was going to say, all those caveats being said,
yes,
what they do in this paper is compare with the same 21-day time point. They compare these 3 vaccine types in different combinations in parallel. And the take-home message is that it looks like this vaccine 9, the Codon pair-optimized vaccine, is superior to the others in virtually every way that they measure. And they have these huge figures with a zillion parts, and they do a lot of work to measure all the different kinds of immune responses. So
it's quite
just
clear.
Wanted to get that summary kind of out there.
Yeah, it is better. It is better in the time frame they've tested. So
I might have missed this, but I'm sorry. Could you please clarify how long the attenuated virus infection lasts for the vaccine?
They give them this vaccine, and then they challenge 21 days later.
Yeah, but are there viruses?
How long does the attenuated vaccine keep replicating?
Exactly.
I don't think I saw that.
They didn't do it. They only looked at the challenge virus to see how long it replicated. Yeah,
Kids, don't you need to know?
That's a darn fine question, Dixon.
Absolutely.
Thank you.
You do need to know.
Maybe I didn't miss that.
Because Dixon, what if it replicated for 10 years? That would be a problem.
Well, that's what the anti-vaxxers are afraid of. But
I don't know.
Well, would that be a problem?
Because polio vaccine, it can reproduce.
Then you'd never be more than 21 days from vaccination.
There you go. Yellow fever vaccine is, I don't know how long that lasts, but it's more than 3 weeks.
Well, polio you can excrete polio vaccine for weeks to months.
Right.
It
depends on your immune system, right?
Everybody's different.
All right.
So we immunize hamsters with, the Codon deoptimized and then the other 2 vaccines. All of these prevent weight loss in hamsters.
Which is the normal way you measure how they're responding to the infection, by the way.
None of them completely prevented infection, but as measured by looking at viral RNA, but the codon number 9 reduced replicating virus to undetectable levels 2 days post challenge, and then when they boost, it gets even better for everybody, including number 9. But the number 9 reduces replicating virus to below detectable levels at 21 days. 2 days post-challenge, which is 21 days post-immunization. So we're still at an early time when there's a lot of antibodies.
One thing I would love to see here is that they are doing their infectious attenuated virus as an intranasal delivery.
Right.
And they're doing everything else as an intramuscular.
Correct.
Yes.
And so it would be, and so they claim a lot that an infectious attenuated virus is the answer.
What would you like them
And
to do, Brianne? Tell me.
I want to know, could they do the infectious attenuated virus in a parallel administration? Could they do that one intramuscularly as well?
Yeah, sure.
And show me whether this is the infectious IAV or whether this is the mucosal delivery that is the real benefit.
So then
here.
why don't we also do the other two vaccines intranasally?
Right.
I mean, they're not replicating, but just put them in there, see what happens, right?
Are all these hamsters inbred?
Yeah, they're in
Okay, so
they're in
what I
loaves
would
of
like to
bread,
see-
Dixon.
I would like to see a wild, quote unquote, wild population of hamsters do the same thing, because that's what we are. That's the next experiment.
Nobody does that.
Nobody does that. But
People
they
are
really
going to get this in
should do
phase
that.
1, Dixon. That's it.
I understand.
We'll eventually get it. Then they look at lung damage by taking out the lungs and slicing them up and looking at them under a microscope. It's called histopathology. This number 9, most efficient at preventing inflammation and pneumonia, which you could see in the microscope, cells coming into the lung and so forth. The other vaccination types, more pathology in the lung, and the prime boost with number 9 was great at protecting the lung. And interestingly, the number 9 was really good at preventing inflammation and fewer monocytic macrophages coming in. I presume because there's less infection, Brianne, right? So you're getting less recruitment, right?
I guess, but I was very curious to think about why exactly this was.
Also, interferon responses were downregulated in animals immunized with. And the only thing I could think of was that there's less viral load. So, right?
I mean, less viral load, but also if you're using a full virus, you do have some of those proteins that are inhibiting.
Oh yeah, it could
the
be
interferon
too.
Response.
Yeah, but it depends if that, that would be transient. So I don't know if this 21 days the vaccine number 9 is still replicating or not. Anyway, we're discussing this, folks, because there's a blunting of immune and inflammatory responses with this vaccine and not with the others. So we're trying to figure out why that would be. They could also see RNA throughout the lungs by in situ hybridization, a technique where you take a section and you add nucleic acid and ask where is there viral RNA in the lung sections. And they see that in animals, even immunized with the other vaccines, you can see viral RNA throughout the lung, even though they don't have a lot of pathology. But in the number 9 animals, they could barely detect RNA. So it's very good at preventing an infection. How about antibody responses? So they collect sera, they do neutralization assays, they ask if the sera blocks infection. So they look at a variety of variants here, and they notice that the sera from hamsters who get number 9 can neutralize a lot of different, but not only ancestral, because remember, this is Delta-based, right? The spike is, this virus is Delta-based, and it neutralizes ancestral as well as forward Beta, Delta, and Omicron BA1, although it's reduced with Omicron. So they make more neutralizing antibodies and broader neutralizing antibodies. And of course, if you, in the number 9 vaccinated hamsters, they make antibodies against not just spike, but other viral proteins.
Yep.
Because they're making them all, right? And that's not the case with the other vaccines. And I guess that would help with T cells, right?
It would. It would give you more T cell epitopes as well.
They're not going to be making neutralizing antibodies, at least most likely. And then they did a number of, what do you call those plots? TSNEs, right?
Yes.
Where they look at all the immune cell populations that are induced by these various immunization routines. They could see differences. Let's see, what can I tell you that's interesting? Yeah, so they can see adaptive cellular responses in the blood after immunization. And the number 9 is really good at inducing these cells of various types. They can see B cells. They can see cells with memory signatures, so they're looking at gene expression, and they say these look like memory cells. They look at both B and T memory cells, and the number 9 seems to be really good at doing that. And on challenge, the activation of these memory cells is best with animals that have gotten number 9. Number 9. They also find that, so you can usually find circulating memory T cells, but what you really want are memory T cells in the organ, right? The lung, the respiratory tract, and only number 9 does that because it's infecting it, right? Tissue resident memory T cells. A lot of pretty picture here, right?
Yeah. A lot of very, very involved figures to dig through.
Mm-hmm.
And
So
this is
Vincent,
open access, so if you want a good paper to dig into, go for it.
right. In the light of what we now know about SARS-CoV-2 infection with regards to cardiac involvement and brain involvement and other organs besides the lungs, are you still enthusiastic about attenuated virus vaccine?
So you're wondering if it will cause issues? Well,
I would.
I don't know, because
I don't want a virus.
I don't know the
infecting
answer.
All of my cells.
I understand, but you would do. I don't know what kind of preclinical work you would do to address that, right?
We
Because
don't have markers for those people.
Because the myocarditis with the mRNA vaccines was not picked up in preclinical or even in the clinical trials because the numbers were too small, right?
Yeah,
Right, but
and
they're not
do
small
we
anymore.
and we probably don't know that the hamster is, or any model is
a
going
good
to be a
model.
great
That's
model for this.
correct, but-
Hamster is a great model for looking at respiratory
Yes.
issues, which is why they're doing hamster here. But
That's right.
that doesn't
That's
mean
right.
that hamster
Yeah.
is going to be a model to tell you those things.
Could you
Yeah, well, they
imagine
would-
a vaccine like this that's missing an element but still allows it to replicate so that it does no harm to the host?
That's tough to do. We don't know.
No, it's tough to do, but it's
I
not
mean, that's why
impossible.
codon deoptimization is attractive because it reduces the
I hear you.
pathogenicity, right?
That's the thought.
In addition to testing this in other animals, you could test it in organoids.
Yes.
See how well this replicates in a human heart organoid.
That's right.
Ideally, from a patient who had, make some stem cells from a patient who had severe COVID.
I don't know. I don't know if replication is going to tell you that. I just don't know.
Maybe
It's
not.
a good point, though, because you don't know.
You don't know.
You're not really going to know until you get this into a lot of people if there's some rare effect. And as I said, the human population at this point has got a whole lot of immunological thoughts about this virus already, and that's going to affect. It may even affect how effective this vaccine is. If you've been vaccinated and infected, and you've got some IgA kicking around, and now you sniff number 9, your immune system may just say, "Oh, this again," and it's gone,
By the way,
and you don't get any
boost.
boost.
That's not love potion number 9.
When you do the phase 1, and then you do the phase 2 and 3, you will know if there are any major, frequent, severe side effects other than the usual pain at the injection site and so forth. But you will not know if something is 1 in 100,000 or 200,000 from that. You will only know that when you put it into people.
That's right.
And if that bothers you, well, it's a problem, right? It doesn't bother me. I'm willing to take the risk because I like the vaccines. I think they're really good.
Yeah.
But you will never find, as Paul Offit says, you will never find any biological that is free of any effects.
Correct.
Other than the ones they are intended to have, right?
I think that's one place where knowing if this is the infectious attenuated vaccine or the mucosal delivery could be really important.
Yeah,
that's right.
Yes,
No, you're
if you don't need to
absolutely right.
replicate, then you don't need to have that in it. Yeah,
That's right. That's right.
The
Or a
last part of
temperature
this
sensor.
is measuring IgA in the mucosa. So they do nasal washes and measure IgA, and they can find IgA positive lymphocytes. And after challenge, right? So these animals who have been vaccinated are making IgA, and they have, of course, reduced viral RNA in the nasal wash as a consequence, presumably, of this IgA. Now, Brianne, I think we talked about this before. Do you get memory IgA B cells there resident in the mucosa, or do they go somewhere else?
You do get some. I don't know the specifics of them in the nasal mucosa, but I know of other mucosal sites where you have resident memory cells.
Okay, because that's what you'd like to
Yes.
do. And this study doesn't really address that functionally, right? They don't
No, not at all.
say if we challenge them 3 months later.
Right.
Will we
all they
see
say is
IgA?
that you get more IgA in the nasal cavity.
And they did do the next step in this experiment, an infection, a transmission experiment.
Oh yes, they did.
So they took vaccinated hamsters, infected. You infect one, you put it into a cage with an uninfected one, see what happens. And this vaccine, at least if you are within that 21-day window, does seem to reduce transmission significantly. It's not a huge experiment, but I think it's pretty interesting.
They put that in as one of the caveats, is that this is really just a preliminary study.
Yes.
The N is 3 for each group.
Yeah.
It's a supplemental figure. It's not in the
Yes.
main part of the paper.
So they put the animals in the same cage, right? So we don't know how it's being transmitted. You know, they could be playing with each other, which hamsters do, and it's not necessarily droplet transmission, but it does tell you that you've reduced the shedding enough so that you don't get, or you blunt the transmission. That's okay. But you know, if you wanted to do this properly, you should do an airborne transmission experiment where you put them in separate cages, right? Let the airflow past them. But I would say that, and this is the number 9 did this better than the other vaccines, right? That's clear.
Yes.
Which makes
There is
sense.
a clear difference.
But I would say if you waited a few months, you would probably not see as much of an impact on transmission, because then you're depending on a memory recall, which is going to be a bit delayed, right? So I was talking to Theodora Hatsuanu about this, and I said, you really need to wait 6 months and challenge the mice. And she said, then we'll never get our grants funded if we tell them we're waiting 6 months.
It would also be interesting to do another type of transmission experiment where you give the vaccine and then put that hamster in the cage with an unvaccinated hamster and see if the vaccine is transmissible.
Sure, because certainly OPV is, right?
Yeah.
The only other question that I have at this point is, I don't know the lifespan of a hamster.
Yes.
So how long?
It's at least a year, I think.
You may not be able to wait too long before you...
Yeah, of course. It's not 2 to 3 years.
Right. Ask any little kid, they'll tell you.
So you could wait 3-4 months. What you would like to do is do an experiment, see when the antibody levels and the T cell levels are contracting, right? And get the baseline.
Yeah,
and then I guarantee you, if you challenge them, they're going to still transmit for a period of time.
Right.
I'm sure.
Yep.
So that's my original point here, is that don't tout this as preventing transmission, because you're doing it at periods when antibodies are so high, and T cells as well, and everything else.
So in virology, let me just preface this by saying that in parasitic diseases that are eukaryotic cells,
Yes,
you can select for temperature-sensitive mutants.
that's right. And that's what
Can you
flu
select
mist
for
is.
temperature-sensitive mutants in virology also?
Yeah, that's what flu mist is, Dixon.
So if you can, then you could probably engineer a attenuated infectious SARS-CoV-19 to replicate in the skin.
Flu mist is temperature sensitive.
It's
Good.
cold sensitive, actually.
It's temperature sensitive and
Oh,
cold
that's right.
adapted.
That's
It-
right. Yeah.
It
So it
only
goes-
replicates under lower temperature conditions.
Yeah, it won't go down into the lung. TS and CA.
Right. So
Now I put
This
my brain.
would-
Yeah, it's here. Too high up. So
This
yeah,
would be a good
that's an
candidate
approach.
for
But
that,
flu
a good
mist is
candidate for that.
no big deal, except the delivery. It's that's great. So that needs some improvement. What I wanted to mention here, this is very cool. In the last paragraph, they say this virus, number 9, is BSL-2.
Yes.
Yes.
As a result of this work.
This is great.
yeah.
The German
Yeah.
authority downgraded it, which is great, because now more. I mean, think about it. How would you develop these vaccines at BSL-3? It would be really difficult. I mean, you could
Yeah.
do it, but
Yeah, you don't want to build a BSL-3 manufacturing facility and you're not going to give a BSL-3 agent as a vaccine because, I mean,
so you have to downgrade
if you
it.
needed that much containment, you shouldn't be shooting it up people's noses, right?
So you have to downgrade it if you want to make SARS-CoV-2 infectious attenuated vaccines, basically, right?
Yeah.
That's very interesting. I found that really interesting. I didn't even think about that.
That's a big and important step. And I assume they're going ahead with this and going to do additional animal trials and hope to get it into humans.
So despite my reservations, I do think we should try this.
I do think they should proceed, yeah.
But as you said, I don't know if in this point in the pandemic, if it could be tested, right?
Yeah, I think the thing to do with this would be to look at, basically look at antibody responses, try to find people who haven't been infected yet, which is going to be tricky, but they're probably still out there. Well,
in some countries, for sure.
I have not knowingly been infected,
right, yeah.
but I've had a couple of colds since the, so, yeah, and you could see if you get mucosal immunity, you could look at markers and say, okay, we're getting a robust response, we're not getting side effects that we didn't expect, and scale it up that way. And I do see a lot of potential for this in the ongoing management of SARS-CoV-2 because we're in the endemic phase now. And every child who's born is going to get exposed or is going to get vaccinated in some order. Speaking from my experience as a parent, it's a whole lot easier to get a kid a nasal vaccine than it is to get them an IM vaccine. So this does have a lot of potential.
So it's just a matter of whether the companies are willing to invest.
Yes.
If they see a market for it, right? Because right now we have a lot of ensconced vaccines, and it's hard to penetrate that market with those already. And that's what they're looking at, Alan. That's all they're looking at.
This one has a major selling point just in the delivery mechanism.
Well, hopefully they'll see it and they'll say,
flu vaccines had been around for decades when flu mist came along, and flu mist is a thing. It's
used, yeah.
It's used, and a company makes it and sells it at presumably some profit that's keeping them afloat. So,
I agree, but we'll see what happens.
Yeah,
it's a lot of people are talking about the difficulty in moving forward with new vaccine candidates, but I think you could find a lot of people who are not infected in Africa. But the ethics of going there to do your trial is another thing, right?
I think you could find people who are not infected in many places.
Do
you think you could find them in the U.S.?
Not infected or vaccinated.
Yeah, or you're not going to find people who haven't been infected or vaccinated. That's going to be
very hard. Yeah, very hard. Because that's another issue. If you want to do a clinical trial, if you have to struggle to find people to enroll, it's difficult to do.
But I think you could do this even in people who've gotten 3 shots and had the virus. If it's been more than 6 months since their last exposure to it, you could look at antibody levels and say we're stimulating an immune response, and then you could take it into younger populations who haven't been exposed, and potentially roll it out that way. I do see this primarily as something that would be a pediatric vaccine. And in fact, I kind of see SARS-CoV-2 vaccines in general as something that's going to become a pediatric vaccine.
Since the world, the WHO approved a new OPV without a challenge, without a phase 2-3, right? Because there's without a trial looking for prevention of polio. They did immunological non-inferiority, right? Because it's all you can do. There are not enough cases to do a proper clinical trial. So there is precedent for that. We'll see. I hope it moves forward because I agree that it has some good properties and the no needle is really good.
Yeah.
Time for some picks, Dixon. When are you going to run out of jazz things?
Just at TWiV 1000.
That's going to be the last one?
Ask me for my pick. I have the last pick at TWiV 1000. You asked me to save it, so I did.
I didn't ask you to save it. I don't know. I don't think we're doing picks in 1000, actually.
Oh, come on.
It takes too much time.
This is TWiV. No, mine wouldn't take time at all. That's all right. I'll just tell you. So my jazz project is winding down, and although it will live on forever because all of these are posted, and I love the way you're following up and putting the songs in the show notes also. I think that's fabulous. You're going
I
to end up with
stopped doing
an
that weeks ago.
amazing record
It's
collection.
too much work.
At any rate, I'm picking conductor composers now, and so I'm finished with the instruments. Gil Evans was an amazing person. He worked almost for free. He died a pauper. He made no money. He invested all of his home money in the studio time to hire the best musicians to come in and play. And Sketches of Spain and Porgy and Bess feature Miles Davis and an all-star orchestra. And the guy was amazing. And then he died. His music lives on, though. These are just amazing efforts, and the the scores are just fabulous. Quincy Jones is best known for his association with Michael Jackson, but before that, he was associated with people like Count Basie and Earl Garner, and there were a lot of other people that he associated with. And I actually had a pleasure of meeting him once and talking to him at a TED talk that we were both present at. And his music covers the waterfront of popular jazz and sometimes even classical music. And then finally, there's Duke Ellington. I don't know what to do with him. Duke Ellington was so prolific in so many directions that the only fault he ever had was smoking. I think I mentioned the fact that Duke Ellington ended up dying where Vincent and I worked at Columbia Presbyterian Hospital, died of a lung sarcoma induced by smoking. And otherwise, his compositions were complex. They were often based in classical music, and he encouraged musicianship. And the reason that he didn't make a lot of money was that he said that my gentleman orchestra has a lot of mouths to feed. And so he ended up paying them what they were worth, and he ended up taking almost none of that money for himself. So he and Gil Evans have that in common. My current all-time favorite that I could listen to on Sunday morning with a cup of coffee and a bagel, or perhaps a well-done piece of toast and some nice salty butter and stuff. Antonio Carlos Jobim is by far the most soothing, gentle, wonderful, and I highly recommend if you really want to escape just for a moment, try The Waters of March. And we just finished with The Waters of March. That is, for me, the quintessential bossa nova song. Of course, The Girl from Ipanema is popular, but almost nobody knows this, The Waters of March. If you listen to it, you will be astounded at the imagery that pops into your head as the words are spouted out. It's almost like a free association of springtime. And it really is quite wondrous. So we're heading up to the goat. I'm going to announce my goat at the TWiV 1000 meeting.
Okay, you can announce your goat because we're not doing picks, right, guys? Isn't that right?
I think we're not. Yeah, I wasn't planning on doing picks at TWiV 1000.
Yeah, but it's a live event. There's no time.
Yeah,
I'll save it for 1001 then.
That's a good idea. Yeah.
And the goat will be behind the second door.
Yes, that's correct. For 30 points, you get the goat. That's right.
Brianne, what do you have for us?
I have a New York Times article. It's referencing a Cell paper that came out, I think it was last week or earlier this week. And that Cell paper got quite a bit of press. The New York Times article in particular I like because the title is funny, and there are a lot of parts in the article that are funny. This is sort of a pun masterpiece in a lot of places. So this article is called "This is What It Sounds Like When Plants Cry." And the paper in Cell was looking at sounds that were produced by plants when they had different types of injury. And they found that different types of injuries led to different sounds being made by the plants, and that machine learning program could actually distinguish 70% of the time what type of injury had happened to the plant. And there were lots of takes about, plant sounds of distress and things like that. And this article is it does such a great job. I have some questions scientifically about the whole idea. I'm not sure that if I break my leg, we would call the snapping noise a cry from my leg. And
it's very hard to tell if these sounds are volitional. I saw the hype around it, and I looked at the work, and I said,
yeah.
okay.
And does anything actually hear them and respond to them in different ways? That's not really clear. They think that this is coming from cavitation of changing in gas levels in the xylem and phloem, but this article explains the whole story in just the best way. Yes. So you can hear the sounds of the plants.
Interesting. Snapping of bones. Wow, that was pretty graphic.
Well,
How about
somebody mentions it in one of the articles about
Joni Mitchell's song, "The Hissing of Summer Grass"?
Yep. It's very evocative, right?
Very
It's
evocative.
good.
Very evocative.
Kathy, what do you have for us?
I have a book that a friend of mine told about. She said, I am binge reading it. I can't put it down. And I got it, and it's read by the author. And I binge listened to it. I had to stop listening to TWiV and any other podcast so that I could complete this book. It's entitled, I Know Who You Are, How an Amateur DNA Sleuth Unmasked the Golden State Killer and Changed Crime Fighting Forever. It's by Barbara Ray Venter. And she does a really deep dive explanation genetically into how this all came about. It's a field called investigative genetic genealogy. She started out just doing genetic genealogy, and I put in the wiki link about her because that tells you some things about her and the book. But this is all using really high-tech analysis of the database, the genetic databases. And there's multiple criminals that she has stories about that she helps figure out, in addition to helping, I think she was helping somebody figure out who their parents were, and then that led to something. You just can't put it down. It's just amazing. She also talks about, there's some interesting reveals about her own family, and I just can't recommend it highly enough. So I Know Who You Are, how an amateur DNA sleuth. So hardly an amateur DNA sleuth. She got a PhD at UC San Diego, was a faculty member at UTMB, and then she went to law school and started doing, I think it was patent law for biotech companies. And her last name is Ray Venter, and I just today, by reading the Wikipedia article, found out that she was married for a while to Craig Venter.
Craig Venter, yes.
gonna
Barbara
ask
Ray
that
Venter
question.
is
Yes, but I had not picked up on that clue.
She
But
reinvented herself.
anyway,
Yes.
what I started to say and distracted myself is that the Golden State Killer went unfound for decades, and basically in something like 67 days from when they started using this technique, they figured out who it was. And then the databases like 23andMe and so forth made people opt in if they wanted their data to be available. And so that severely reduced the amount of data that could be traced. And so she said, I would be unable to do that same identification now. But the databases are kind of repopulating, and so it's getting better. But yeah, and she talks about women in science. And did you know that a hair doesn't have to have a root shaft in order to give you DNA information? That was something I learned. Just amazing. So highly recommend.
Cool.
Neat.
That sounds good. Alan, what do you have for us?
I have a little virtual tour people can take online of a place they're highly unlikely to ever be able to visit in person. I certainly don't plan to go here myself. This is the Svalbard Global Seed Vault, and I know Dixon is itching to take a look at this.
I've already done that, Alan. I know the guy who invented it.
Yeah, so this is
Carrie Fowler, and his name is Carrie Fowler.
You can go there online and see the place for yourself now. So this is the sometimes called Doomsday Vault.
That's right.
The idea is, there are a lot of crops that we depend on. There are a lot of plants and seeds that we need. And it could happen that some of these heirloom strains of things get lost. And then they're gone. They're no longer available to humanity. So this vault was created a number of years ago to store samples of the world's seeds. And they built it into... it's built into a hillside in Norway?
Yes.
Yeah, Norway.
Svalbard, yes.
Svalbard, which is not convenient to anything in Norway, and it's not supposed to be, because this is not a place that you go and tour. It's just a hole in the side of a mountain with a big door, and they store the seeds in there in freezing conditions, so that they'll keep for a long time, and you can go in and look around in this virtual tour. It's very cool.
Yes.
It's a lot of space.
It's a lot of space because they've got a lot of seeds to hang on to.
There's a caveat to this one, unfortunately, yes. And you can mention that if you want, Alan. I don't know if you saw the latest Nova, but it was all about the melting of the permafrost.
Yes.
So this falls within that purview, and
this site may not be quite as stable as they had hoped.
It's not, and it suffered a flood once.
Yeah.
But I met the guy who actually got the money from the Norwegian government to fund this thing. He was married to a Norwegian, a guy from Kentucky, of all places, and Carrie Fowler was his name. We had a wonderful week together in Seoul, Korea, of all places. And he told me lots of wonderful stories about this vault, and he was there to talk about it as well. And it's interesting because South Korea, after hearing both he speak and me speak, I don't know if that was said correctly, but it doesn't really matter. They established a vertical farm and a food seed bank. In 2 years after we spoke, they had it up and running. South Korea. So that tells you something. You never know who you're talking to, and you never know who's listening. But he was kind of, how should I say it? He was skeptical of his own invention, basically, because every country needs one of these things, not just Norway.
Sure. Norway's, I think, is intended to be sort of a vault of last resort.
It's a worldwide thing. It is, but it's...
Kathy.
So Svalbard is where Longyearbyen is, which I think is the northernmost city or town or whatever metropolis it would qualify for. And that's where someone went and looked for influenza samples in bodies.
Correct.
Yes.
We've talked about Longyearbyen on a past TWiV at some point.
These chambers, I'm going through, many of them are empty.
Yes, they've got a lot of space, which is a good thing, but they're filling up some of them.
I think you should put the TWiV archives in there. That's just kidding. They're in my basement. I'm going to bring them here one day. Actually, the TWiV archive exists. All the episodes are duplicated on 2 spinning hard drives. And I should really keep one here and one somewhere else, right?
Yeah.
In case one place burns down. But they're very heavy.
Floods.
Yeah. They're very heavy. I have over 50 of them.
Of course, the sound waves are going on forever, right?
Who knows? Yeah, anyway, that's very cool. Neat.
Yeah.
The space is great. I wonder if you could stand to work in there. I guess it would be claustrophobic.
It would be cold.
Cold too.
Yeah, because it's freezer temperature. It's like minus 20 C.
That's right.
So they put it in this hillside so that they could economically keep it at that temperature.
I see.
That's their thought. But what happens when that melts?
Well, yes.
Not if. When.
My pick is a story in Atlantic by Catherine Wu. I think this may have been picked before, but it doesn't matter because it is worth reading, and it's the kind of thing where Catherine seems to get it. I actually just emailed her this week and invited her onto TWiV, and she's very excited to join us.
Yay!
Excellent.
Great.
So
Awesome.
We'll find a common
Very
date.
Cool. Very cool.
We're asking the impossible of vaccines. Complete protection against infection has long been hailed as the holy grail of vaccination. It might simply be unachievable. Catherine Wu. And notice she says it might be unachievable. That means it's not been achieved. Okay? And
Right.
she's right. And she talks to a number of people who say, we don't even know how to figure out if they do. I mean, think about what experiment you'd have to do. You would have to vaccinate people and then test them regularly for a while to see if they ever got infected. And that's hard to do. And nobody's ever done that. So I like the story because she talks to people and explores this issue. And one of the cool things that comes up, she says, well, you know, people say the papilloma vaccines are sterilizing. Well, it turns out that they prevent 99.95% of cancers. And so people assume they're sterilizing, but it's never been shown. In fact, may not be. They may
Sterilizing is not the word to use there, though.
Well, there it's not a good word
Right.
to use. Block infection entirely.
Exactly.
So people are saying that this is one of the vaccines that does block infection, and it doesn't. It blocks cancer really, really, really well, but not infection. Anyway, good article. She's got a whole bunch of good articles there, and we'll have her on. We can chat about them. Should be fun. She's got a PhD in immunology from Harvard.
Oh, nice.
So, it's hard to write about science, and if you have done science, it makes it a lot easier, right?
Yes.
So I taught 2 lectures last week in Jonathan Weiner's science writing course in the journalism program at Columbia.
Mm.
Jonathan is a Pulitzer Prize winner for writing The Beak and the Finch, and he's been on TWiV a couple of times. And they have 10 people in the class who want to do different kinds of writing, some of them science. None of them have any science background whatsoever.
Naturally.
None. I mean, barely a course in college. And I gave Jonathan said, give us a primer lecture where you give us some science background and then talk about viruses in another lecture. So tell us what we need to know. And they're 3-hour classes, right? Half of them were asleep in the first class. And this is a tiny room, right? Where I'm right in front of them.
How many students?
Did you clap at him and say, wake up?
That's
Try and
right.
raise my voice.
The first thing is to stay awake. Then the next thing is
Because it was, your bodies are built of these things, and this is what they look like, and this is what DNA looks like, and all things I thought were really cool, but they could care less about. And
Right.
and then when I talked about viruses, they all stayed awake because they're asking questions every minute, right? So I
Right.
told Jonathan, next year we have to do something different because it's not good to talk to them for 3 hours. They're not going to remember any of it, right? Even
How
though
many
I thought it
students,
was. How many students? I did 10 students. Right?
oh, just 10. Oh.
When I taught at NYU's journalism school, I did a section in there. In the science and environmental reporting program, they have a course where they teach science to aspiring science journalists. That they review science.
Yeah.
Because most of these people come from some kind of a science background,
Yeah.
but then they're potentially going to be reporting on stories outside their field, and they need to have, make sure that their understanding is sufficiently crystallized on all these subjects. So I did molecular biology in about 4 weeks or something. First lecture, I figured, okay, I'm going to keep these people awake by covering the material briskly. And so I had a lecture that went from a carbon atom to a cell. And I managed to keep them awake. They were trying to keep up, and I think some of it got retained, but then we moved on to things that were more discussion prone.
Yeah.
To get, how does molecular biology matter in the world, and what kind of stories are you going to be covering about it? And that's where people actually get engaged to to the topic.
Yeah.
The best course I ever gave like that was to pharmacy students of pharmacy and pharmacists, practicing pharmacists that were taking an adult education course at night. And I started out by drawing 3 chemical formula on the blackboard. The first one was iron, 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 iron.
A Ferris wheel.
Alan, you're a good pharmacist, good boy. And then the next one was CH2O. And by the way, these people were panicking at this point. And then finally, I drew the benzene ring connected in so many different ways you couldn't possibly imagine what it was, and then put 2 OH groups on it.
Dihydroxy
And they
chicken
thought,
wire.
yes, you are correct for 30 points. You go to the head of your class. The other one was seawater. So I said, this is all the chemistry I'm going to talk about.
Seawater.
And then they put down their pens and started to
good.
laugh. They came up and shook my head afterwards.
Like that.
It was a lot of fun, actually, a lot of fun.
Yeah, I have a student in my virology class who is also in that journalism writing class. She's
Really? That's a good idea.
an older student who has written and done journalism for 20 years. And
Oh,
I
wow.
said, what are you taking my course for? She said, because virus writing by the press is abysmal, and I want to see if I can do it better. So
Here, here.
she's taking both courses,
Excellent.
and I admire that very much. But, getting a job is not easy to do that.
How do you get them
I actually
to
gave
share?
her your email, Alan. I said, you should talk to Alan because he can give you good advice.
Right.
Sorry,
So Vincent,
Dixon.
how do you get the students to share their article with you before they publish it? So you can correct all the mistakes.
What article?
The ones that are writing.
No, you're not going to have time to do that. That's
Well, that's the problem, isn't it?
you need to. No. Vincent needs to be the vaccine. They need to be immunized against screwing up to some extent, or understand the thought process that they need to ask the right questions to get the answers for the
But
story.
how would you even know if they understood you?
Well, you can tell by their
Only
questions.
when you read the article would you know.
No, but
Right,
they
but
ask
you
the questions.
and you can do. I don't know if your class does this, but we had in the SHRP program, we certainly had writing assignments.
Yeah, of course.
And I would have them cover things.
No, my
Margarita
lectures,
Hathaway.
Dixon, are just background. They don't write anything for me, but they do write for Jonathan
No,
throughout
I
the
understand.
course. Yeah.
I understand.
But they ask, I can tell if they're asking questions that they're getting it, right?
Yeah.
The virus part, they got great questions. And the molecular biology part,
I'm going to tell you,
very
2
little.
days later, they'll forget all of that.
That's
all
the
of
third time, Brienne, right?
I know. This never happens, and I guess today I'm very still.
We have there's a bathroom here in the incubator down the hall where the light goes out after 5 minutes.
That's not a good thing.
Yeah. All right. That's TWiV 999. You can find the show notes.
I do have one thing I want to add.
Yes,
just
sir.
real quickly for those pilots who are listening and the airline pilots who are tearing their hair out. How did he not know? The typical approach speeds for an Airbus A320 I looked up. They're in the ballpark of 130, 140 knots, which
Right.
is faster than the cruising speed of most of the airplanes I have ever flown. So,
Okay. Right. Yeah. Well, they may not wait. They may stop the recording
they
and,
may stop and write in.
and
yes.
write in immediately, which is
So
fine.
I
It's no
Already
problem.
got it.
All right. TWiV 999. Oh, this is funny because our vaccine was number 9.
Number
right?
9.
That's
yes.
right.
You can find the show notes at microbe.tv/TWiV. You can send us a question, a comment, or a pic, TWiV@microbe.tv. And if you enjoy our work, if you've liked these 999 episodes, we would love your support. We are trying a model where we don't use ads and we depend on your contributions to do this kind of science discussion. Microbe.tv/contribute. Dixon Despommier, Tricanella.org, thelivingriver.org. Thank you, Dixon.
Thumbs up. Good show.
Thumbs up.
Can hardly wait for the next one.
Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Alan Dove, turbidplaque.com, alandove.com. Thank you, Alan.
Thank you. It's always a pleasure.
Brienne Barker's at Drew University, BioProf Barker on Twitter. Thanks, Brienne.
Thanks. I learned a lot.
I'm Vincent Racaniello. You can find me at virology.ws. I would like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Jolene for the timestamps, and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.